Joint disease affects more than 50 million people in the United States, and we spend a staggering $130 billion every year in treatment costs. A joint injury can often be career-ending for both human as well as equine athletes. So why is this such a major problem? It's because the joint cartilage is an easy to damage and incredibly hard to repair tissue. It forms a smooth cushion where the bone ends meet at the joints. And during disease or injury, this tissue gets worn away, exposing the bone underneath. So when the joints move, the bone ends rub against each other, causing friction, pain, and immobility. Research has tried to recreate the amazing properties of this joint cartilage for decades with limited success. In my PhD research, I'm looking into a population of cells within the joint called the interzone, from which the joint cartilage develops in the embryo. When I put these cells in a bone fracture, the fracture heals completely, whereas in a site that's far removed from the skeleton, the cells remain as such, doing nothing. So it's like planting tulips here in Kentucky. They do great, but if you try to grow them in somewhere like Arizona, it just doesn't do as well. The climate doesn't support it. So there is something unique about the environment that these cells live in that plays a big role in the repair process that we see. So how do the interzone cells communicate and interact with their environment? Are they being told what to do, or are they the master switch that regulates all the other cells? Is there a signaling going on between the interzone cells and the skeletal cells at the cut ends of the bone? These are some of the questions that I'm trying to answer in my PhD research. The data from my experiments so far shows that the skeletal cells do produce signals that have an effect on interzone cells. In the next set of experiments, I'm trying to narrow down the specific signaling molecules that could be at play here. This study will provide valuable information on the functionality of interzone cells that could translate into its potential use as a cell for restoring cartilage to its normal form and function. Thank you.